And it was August Weismann, a distinguished German biologist, who realized that what was really continuous was eggs and sperm, what he called germ plasm. And if you like, you can think of us as just mere toadstools, spore-carrying bodies to exist only to produce the next generation. But of course, this doesn't really explain what is continuous. And the next great advance came when people saw chromosomes, which was a really accidental discovery based on the invention of aniline dyes. And once people put aniline dyes on cells, they saw these things. And this is one of the first drawings of chromosomes inside cells. And you can see that there's this mysterious thing, which we would now call the mitotic spindle, which is going to be used, as we'll see shortly, to separate those chromosomes. But at the time, there they were, and people could see that there was something funny going on them when cells divided, but they didn't really know what they were. But it took another 20 years to realize just how important they were. And I wish I had time to tell you the beautiful, simple experiments that this man, Theodore Boveri, did in order to prove the wonderful statement here. I strongly recommend, if you have any interest in this at all, to read his 1902 paper, which was originally written in German, which has been translated into English, and you can find on the web if you type Boveri 1902 into the web. Only one possibility remains. It's a beautiful, tightly argued paper. Namely, not a definite number, but a definite combination of chromosomes is essential for normal development. And this means that the individual chromosomes must possess different qualities. There's something really important in chromosomes which lead to the development. A cat has cat chromosomes, human has human chromosomes, an oak tree has oak tree chromosomes, and they faithfully record all the instructions to make those organisms. And there is a fantastic mechanism for distributing chromosomes between two daughter cells, and it's called mitosis, from the Greek, I believe, for a thread. You can correct me if I'm wrong about that. And this is something that you have to learn, that there are preparations for cell division, and then we reach a culmination in metaphase where the chromosomes are lined up, then the chromosomes separate and go to two daughter cells, and finally the cells divide into two. And I want to show you now that in a movie made by a young colleague of mine, Mark Petronsky. Look for the blue thing. So we start out with this number of cells, and by the end of the movie, there will be twice that number of cells, and the blue fluorescence is the chromosome. So watch carefully, because it happens in the moment, in the twinkling of an eye. There we go. Boing, 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 boing. It's fantastic. Isn't that great? And what's going on there? Why are these cells dividing to two, and what's that separating? Well, this all depends, as you very well know, of course, or practically all of you, on the famous molecule DNA. And why do cells divide into two? It's because DNA has two strands. One strand goes to one daughter cell, the other strand goes to the other daughter cell. And why is that important? It's important because of the famous central dogma of biology, which I believe is due to my predecessor on this stage, Jim Watson. According to Francis Crick, Jim had it first. And the central dogma of molecular biology is that DNA makes RNA, makes protein. And actually it's protein pretty much that do everything. They're the enzymes, they carry the oxygen, they allow you to see through your lens. You know, this is protein, that is protein. Everything is proteins, virtually. Of course, there are people who will tell you that that's not true, and they like lipids or something like that. But I don't like lipids. They accumulate round your tummy and make you fat. But the only trouble is, and it is true, that really it's the proteins which make cells and the cells which make organisms. And I want to show you just the sheer scale of this. I think this is my favorite display on the Euston Road in the museum of the Medical Museum of the Wellcome Trust. This is a printout of the human genome. It's a library of the entire human genome, which was sequenced successfully about the year 2000, 2001. It's still being refined, actually. So you can see that it comes in 23 volumes. It's written in 4-point Courier, so it's pretty small. And let me show you the page that's open in front of that young woman there. That's what DNA looks like. And that is the instructions for making the beta chain of human hemoglobin, as several of you will have recognized, of course. Now, you'll notice that it isn't actually a terribly good read as far as we're concerned. There are no paragraphs, there are no quotations, there are no full stops, no commas, nothing. Absolutely amazing. And there's an incredible amount of information in there. Exactly how much? I looked up to see how much the complete works, how many characters are there in the complete works of Shakespeare. I could have looked up the Bible, but it happened Shakespeare came up first. Well, it turns out that the complete works of Shakespeare comprises 3,695,990 characters. I don't know who counted that. It must have been a very boring job. The human genome has 3 billion characters in it. So that means that each one of your cells, each one of your 10 to the 13th cells, contains 812 complete works of Shakespeare's worth of information. Isn't that incredible? I mean, absolutely fantastic. And apparently that's what you need to be a human being. Now, people argue about how much of this information is rubbish or junk or, and the general consensus is that only 25,000 of these sequences are actually genes, but probably quite a lot of the stuff in between is important for one reason or another. Otherwise, it wouldn't be there, but we still don't know. That's one of the deep mysteries. So back to cells and cell division. So what happens, and Watson and Crick really realized this quite early on, was that the strands of DNA separate. Remember that Francis Crick's great contribution was the discovery that the two strands run in opposite directions, and that creates great problems here at the replication fork. And actually, quite often during the replication, mistakes are made because it's impossible thermodynamically to make it exactly right. And as Lewis Thomas, you may not be able to read this at the back, but I love this quote, "The capacity to blunder slightly is the real marvel of DNA. Without this special attribute, we would still be anaerobic bacteria and there would be no music." Lovely. So if it wasn't for occasional mistakes in the replication of DNA, we wouldn't be here. Evolution would be impossible. But our job is to try and find out how cells manage to duplicate and then segregate these daughter chromosomes to the two daughter cells, so that every single cell in your body has that 800 complete works of Shakespeare's worth of information. So any cell in your body could potentially become a new human being. And as you probably realize, with the new advances in stem cell technology, it is actually possible to take a cell from a human being and turn it back basically into an egg which can develop into a new human being. I mean, basically all the information is there. It's just very selective, just as when you go to a library, you pull out the books you want to read. So a germ cell can sort of wipe the slate clean and set back the development right to the beginning of time. So you replicate the DNA, you make two daughter chromosomes, and then you pull them apart. One of the great discoveries that was made not so very long ago was that actually these two daughter chromosomes are held together by little elastic bands of proteins which are released. And here's a little movie that I found on the internet. I paid $15 for it, I think. And it's kind of nice. I'll just run it 2 or 3 times. And you can see the chromosomes lining up in the middle of the cell and then being pulled apart by this special thing. It's not quite right, but never mind. So we can formalize that, and my boss, Paul Nurse, made this nice diagram showing the chromosomes being replicated. 1 goes to 2, and then separating so that 1 goes to one daughter cell and the other goes to the other daughter cell, so that each time a cell divides, you have twice as many cells as you had before. And it was in 1953 that people discovered that actually the cell cycle looked like this. It was quite orderly. the replication of chromosomes was distinct from the segregation of chromosomes. And moreover, there are things that people came to call checkpoints. That is to say that this little cycle doesn't just go round and round haphazardly. It goes round only when it's ready to go round. And for example, you can't go on and enter cell division if you haven't completed the replication of your chromosomes. There are mechanisms which detect incompletely replicated DNA. We don't really know what they are. There are better studied mechanisms which find out if the chromosomes are damaged, and if the chromosomes are damaged, the cells won't divide, because the last thing you want to do is pass on incorrect information to the next generation. And it turns out that evolution has evolved some wonderful mechanisms for detecting damaged DNA, as well as repair mechanisms. And I should mention that my boss at Clare Hall Labs, Thomas Lindahl, who is a wonderful scientist, a wonderful boss, was quite rightly awarded this year's Nobel Prize in Chemistry, or share of this year's Nobel Prize in Chemistry, for his work on how DNA suffers damage and how that damage is repaired. I should mention, everybody knows about smoking and cancer, but do you know what the two most deadly enemies of DNA are? It's actually oxygen and water. Now, unfortunately, it's rather hard to evade oxygen and water if you want to be alive. And so the cell, again, has evolved mechanisms for correcting the damage that is done by hydrolytic decay of DNA. So this is quite complicated, you know, and you see there are sort of amazing devices for replicating and segregating chromosomes. So how is all this controlled? Well, my introduction to this came from an interest in protein synthesis and its control. And as already been mentioned, I used to go to Woods Hole, Massachusetts, where there are summer courses, and it's a very nice place. You can see it's entirely surrounded by the sea. It's on a peninsula, a little tiny village with two big labs in it. The Woods Hole Oceanographic Institution, which is mostly run by the US Navy, and the Marine Biological Laboratory, which has been around quite a lot longer and struggles for survival. But the Woods Hole summer courses there are absolutely superb, and if anyone ever gets a possibility of a chance to go there, I would highly recommend that you take it. They run in all kinds of things, from experimental embryology to advanced artificial intelligence and things like that. So I was there to get sea urchin eggs, because sea urchin eggs, when they're fertilized, start making proteins anew, and I wanted to find out how protein synthesis was turned on. And the way you fertilize a sea urchin egg is you get a female sea urchin, and there's a male sea urchin, and you can see the eggs look rather different. The eggs actually are, I'm sorry, these are black and white photographs. The sperm is white and the eggs are deep purple. And if you mix them together, this is what you see. Very quickly, about a minute after mixing them, you see this envelope appear around them. And now if I run this is a movie, you wait about an hour and the cells start to divide. And they divide amazingly synchronously. That's 4, 8, 16 cells. And if you leave them for hours, they will turn into a new baby sea urchin. And as I say, I was interested in what turned on protein synthesis. And I became aware that if you blocked protein synthesis, which is easy with antibiotics, the cells did not divide. So clear take-home message, one or more of these newly synthesized proteins must be essential for cell division. But funnily enough, nobody really paid attention to that, because everybody knew that cells normally have to make new proteins to divide because they have to double in mass. But this is a case where actually they don't double in mass, they halve in mass each time they divide. And that really simple logic was not borne in on it until I thought about it and worked there for quite a few years.